it's very nice that you decided to spend this 1 hour and maybe 15 minutes together with us, and together with Mara Graziani. Mara is at that moment is a PhD candidate in computer science, and officially she is affiliated at the University of Geneva. But he's doing his research in a nice place of Switzerland. You can maybe so she's a really happy person, and the main topic of her interest is improving way in which machine learning is applied in healthcare. In healthcare. And while we were working together with Mara in a project which is called Process, and this is about future large-scale computing systems, and application which is developed by Mara is demanding for such high performance computing systems, which will be in the future. So I'm very grateful to Mara that she decided to join us, and Mara, floor is yours.
Thank you very much. Yeah, it's a pleasure for me to be here. It's always nice to talk about my work and share it with people and engage in discussion. So I'm really happy to be here. Today I will give a global overview on the application of AI to digital pathology, and I will particularly focus on some of the challenges that arise from dealing with this kind of data. That can be like dealing with the computational requirements of this data, the level of heterogeneity, and also the need of interpreting the complex decision-making of AI models used in the context of healthcare and especially of pathology. So we had to deal with these challenges as part of our tasks in the European project process. This project aims at developing computing and data management solutions for highly demanding applications in terms of computational storage requirements. So just to introduce myself before the talk, I'm a third-year PhD student at the University of Geneva in Switzerland, and at the same time, I've been working for the University of Applied Sciences in Basel, Switzerland. That is called SSO Valais, and the main research topic for my PhD is actually on the interpretability of deep learning models for medicine. And so while working on the use case for the process project, I had the chance to train deep learning models on histopathology data, and I had to face up with the challenges that I'm going to talk about during this talk. So the works that I will present are the product of the collaboration over the years with many people. I'm acknowledging some of them, and the pictures here will work with me at SSO. And some of the results that I will present were also possible thanks to the collaboration with the other partners involved in process. So the infrastructure that they built actually gave us to the computing and storage resources that we're using to make the experiments. As we mentioned already, I work in Switzerland in this nice place here, and you might be wondering like where we actually located. So I added like our location here at the bottom left corner. We are halfway between Geneva and Milan, and halfway between Geneva and Zurich. So we're nicely connected a bit everywhere, but we are still like kind of a retreat in the mountain. So, okay, I guess we can move on now from the introductions and I can start the talk. So what is digital pathology? Like maybe some of you might not be familiar with it. So let me please introduce briefly this field. I'm not a pathologist. I come from computer science background, so I would try to say this with other people's words. Wikipedia, for example, identifies as pathology the analysis of tissue, cell, and body fluid samples as the pathology analysis. So this can be defined also in a loose sense as sort of the medicine's ground truth that clarifies and confirms eventual hypotheses that can arise from previous steps in the diagnosis. So let's say if you have some symptoms, I hope not clearly, but if you have some symptoms and these are analyzed to determine whether they are clinically relevant signs or not, then this would motivate a checkup from a radiology impression. And finally, on the basis of these steps, if a pathology diagnosis is required, it means that there was some suspect activity that was found, and the pathology will assess the malignancy of that activity and then plan the treatment strategy. And this is done by analyzing the tissue and the cells at the microscopic level. So this is a summary of the pathology workflow. Tissue specimens are taken from the patient with needles or by surgical removal, and then they are sent to the laboratory. If a full tumor area is removed, then this is cut into slices, and the samples of tissues then undergo a procedure that is called staining. Now, the golden standard of pathology uses two types of stain that are called hematoxylane and eusin, and these stains highlight the cells and nuclei and the tissue structures in the slides. And the tissue is then trapped between two thin glass slides. And the pathologist then analyzes the glass slides at the microscope to determine the tumor grade. A further step was introduced in the last decade to this pipeline that is mostly analogic, and it is the digitalization of the glass slides by scanning them with extremely powerful scanners that can take images at different resolutions to mimic really the different levels of details that can be reached by a microscope. And here I like this example, the example of this picture where you can see the glass slide in the hand of this man, and just behind him there is the digitalization of it into a slide. So digital pathology essentially is the collection of these all of these images for diagnostic purposes. Now, to dig a bit more in the details, I'm showing here a part of a digital slide. And as you see, there are different structures in the image with different cell types. For example, like on the right, there is a red stripe that is actually made of blood cells. And the black mark at the bottom left is showing you that this mark is long 1,000 micrometers. So this gives you a bit of an idea of the actual physical dimension of what is being analyzed. And these images are analyzed, as I was saying, at different levels of resolution. So the pathologist zoom in and zoom out to determine whether there are particular areas in the tissue that correspond to some guidelines for the grading scheme. For breast cancer, for example, pathologists look at three things. They look at the overall structure and organization of the cells. They zoom in and then they look at the nuclei shape and appearance. And then they try to count the number of mitoses, which is a very difficult task that essentially consists of identifying very particular cells with some activity, some specific activity going on. So this scheme is sort of a half qualitative metric that really depends on what the pathologist follows when zooming in on some areas of the images and focusing on some nuclei rather than others. And they start the analysis essentially and they move around the slide at this resolution. So it's very high resolution. So as you can imagine, spending several hours on this kind of images can be very tiring, and it's also time consuming. And as I was saying, this is very subjective. So there is really not clear objectivity in how the analysis is made. And I mean, as a consequence, if we were to take a bunch of pathologists and compare their diagnosis on the same slide, we would find out that very rarely their diagnoses agree. In this study here for breast cancer, for example, each column here represents a patient sample, and the output of a pathologist is represented as a color. So essentially you see that for each column, and that is one image, you have a variety of outputs, meaning that there is very low concordance on what is the actual interpretation of the image. And from this, we can actually think that AI could help to introduce objectivity in the analysis and possibly reduce the time and the fatigue as to pathologists when analyzing these images. So there's this impressive study by MIT here, for example, that has shown that by combining pathologist opinion and deep learning decisions, then the error rate drops significantly. And this motivates the whole work of research in this area. Essentially, we can see that we can achieve great results by combining deep learning models and experts in the field. And this data anyway are quite challenging even for AI. Like there is a high computational demand behind analyzing images with billion of pixels. Each image of these can occupy, for example, between 2 and 3 gigabytes per glass slide. And for some organs, this might require like between 12 and 20 slides to be collected, because like one slide is not enough to actually classify the cancer grade. So if you do the math, this can scale up quickly to large data sets with very important storage requirements. And another problem that could cause a lot of issues when trying to train deep learning models on this data is that there is a lot of heterogeneity. That is due to essentially to the lack of standardization of the acquisition protocols for digitalization and for like the digitalization of the slides. So small shifts in the staining procedures, for example, or in the temperature of the samples, or even the scanner producer may take images with different encoding, and this may cause gaps in the data distributions that then are difficult to realign before training deep learning models. Even more, like manually annotating each piece by identifying each and every abnormal mitotic nuclei as quite a high cost, and this requires several hours of tedious and tiring work. So these were some of the challenges that we actually had to face up in the lab while we were developing our deep learning models for digital pathologies. Just to summarize them again, the unconventional image sizes that are of 1,000 times 1,000 pixels at least, goes up to a billion pixel in one image. Then there is plenty of data being collected, but actually the annotations are scarce. So, and mostly they are high level. So there is not a fine-grained segmentation of the regions containing tumor always available. Then there is the fact that the analysis has to be at multiple resolution because at different scales, different levels of details are visible and different features can be noticed. So this has to be done in a multi-resolution analysis. Another thing is that contextual information matters. So generally, if we look locally at one part in the slide, we know that if there is tumor in there, the surroundings are more likely to also contain tumor. And often this contextual information is forgotten when deep learning models are applied. There is also the fact that very often annotations come in different modalities. So sometimes they could be written reports, sometimes they could be segmentations of the areas, sometimes there could be just a label whether the slide contains or doesn't contain tumor, sometimes there could be annotations segmenting the individual nuclei. And this also goes with the lack of standardization. So there could be different scanners being used and different staining methods. So this would generate domain shifts in the data distributions. Another issue is also that these data are highly imbalanced. So the tumor regions are often needle in a haystack. I will show this later, but essentially it's a very small area that generally has classified as positive, and all the rest has to be classified as negative because it doesn't contain tumor. So yeah, within the process project, we started from one specific application. So we reduced a bit our field and we went to checking just the identification of metastasis in breast cancer, breast lymph nodes. We used a publicly available connection of nearly 3 terabytes of data with 500 images fully annotated, like with schematic annotations of the tumor regions. And these images contain an average of 1 billion pixels each. So as I wanted to underline before, in these images, a single tumor cell may occupy less than 0.008% of the entire image. So you see that what I was saying about finding a needle in the haystack is kind of the problem that we're trying to solve here. We developed a region of interest proposer that is essentially an algorithm that suggests salient regions to look at for the pathologist. This was successfully done in research by following a very specific pipeline that we decided to replicate. Essentially, at training time, random locations are sampled from the gigapixel images, from which then patches at higher resolution level are cropped from the image, from the annotated and the annotated tumor and the non-tumor regions. Then a convolutional neural network is trained with a binary classification task to determine whether each image contains or not some tumor. At testing time, the image is also split into patches or tiles, and these patches are passed to the CNN to build a heat map of the probability of tumor that would determine which areas are the most at risk because they have highly likelihood to contain tumor, a higher likelihood to contain tumor. Now, in this application, we saw these emergence needs for the future that we decided to address while replicating the pipeline. Our first concern was the need for an efficient process that can extract image batches. One bottleneck, for example, in replicating this pipeline is the number of batches that each research center can extract because of computational bounds. So since better performance is generally achieved with a larger number of batches, then we focus on developing an algorithm that is efficient enough and that uses the high computing power of high computing centers to extract millions of batches. Then portability was the second point that we decided to address. We wanted to be able to perform this pipeline on any type of infrastructure without the time step of reinstalling the libraries and matching all the requirements. And this is something that we do often here at SSO, that is working on the evaluation as a service, where the idea is essentially to move the algorithms to the data rather than the opposite. And this would be also useful in the future in the sense of federated learning by moving the algorithms to the different hospitals that could then replicate the pipeline and local with their private data that they cannot share. The third point is that one might actually not need to replicate exactly the same pipeline. So we needed modularity to allow future researchers to actually interchange bits and pieces and reuse only the parts that they needed. And finally, we cared a lot about the interpretability aspect of the application. CNN is often perceived as black boxes by pathologists, so to concretely propose something that would be used for the future, we recognized interpretability as an emerging need. And I mean, as I already mentioned, this is also a main topic of my PhD thesis, and the idea here is to meet the new European regulations in terms of providing justifications for automated outputs. So in concrete, what did we do? What did we develop, and what kind of results and publication did we have as an outcome of the project? We came up with this goal. We wanted to develop a region proposer that could meet the requirements, obviously, of efficiency, portability, and modularity that I just talked about. And we wanted at the same time to be able to explain the decisions in a human-friendly way that could be directly understood and proposed to pathologists. And to do so, we tried to answer questions such as why is the algorithm focusing on some regions rather than others, for example. So we developed an algorithm with interpretability that would highlight a high-grade tumor region and saying this is highlighted as high grade because the cells are larger than the non-tumor average and because the nuclei texture appear vesicular, for example, because it's high in contrast when you compare to the average. So one of the main outcomes of the project was the development of open source software that is efficient and portable and modular. And this was done by developing a 3-layered application where each layer can be independently deployed as a Docker container and can be also interconnected to reconstruct the full automated pipeline of the explainable region proposer. To look in detail at each layer, we have that layer 1, for example, focuses on the data preprocessing and the patch extraction step. So we created masks of normal tissue and tumor tissue. And from there, we implemented the sampling of batches at high resolution that I shown before. And we decided to have both a random sampling, so a random sampling of locations, and both covering densely all the possible locations that could be extracted. And we created an intermediate database of batches where we could store all of our data for training deep learning models. Layer 2 then focuses on the training of deep learning models, and we thought of changing the different networks. So we have a config file essentially that allows you to choose your state-of-the-art deep learning model and train it on your data. And we wanted to implement both the traditional training of deep learning models and the distributed training by using multi-GPUs approaches and scaling the training on the computational resources that we had. And the last layer was the performance boosting and interpretability. So we cared about generating visualizations and analyzing the importance of individual features and answering these why questions to satisfy the data protection regulation. So for example, here, the first Docker container that we developed is the patch extractor that extracts patches from the slides at the desired resolution level. And we first did random sampling. So we generated locations and we're extracting patches. But then since we had resources within the project, we were able to extract actually one patch for each location in the image. And we ended up by densely cover the image. Just to mention and to give an idea, with this, we ended up generating a data set of 43 million images. That is almost 43 times the ImageNet data set that is known in the computer vision community as one of the largest image data sets. So one thing we observed thanks to this is that we could almost cover a linear speed up under the condition of having enough CPU computing cores for extracting images at high resolution. And this means that our sampling can essentially deal with increasing number of patient data being collected within the medical community. And this would be possible by essentially parallelizing on supercomputing centers that can provide thousands of CPU cores, the patch extraction part. The second layer of our application consisted of training different neural networks, as I was saying, that could identify tumor and non-tumor patches. And one main issue that we had to deal with here was the heterogeneity in the data, because the data that we used were collected in different centers, and this affected a bit the classification. So we actually did not focus our models. We did not focus on optimizing our models to achieve challenge level performances, fine-tuning every bit and byte of our models, because this wasn't our main objective within the project. But what we tried to do actually was to expand the range of data sets and the range of heterogeneity that could be taken into account while analyzing this data, and focusing on the generalization of our model. And we achieved a nice area under the ROC curve of 0.85 that still compares well with other papers in the domain that were not optimized to challenge level. To do this, we also, as I was saying, we used Harvard library developed by Uber to parallelize network training over multiple GPUs. And this was nice because we could actually check if we could improve the efficiency of network training by parallelizing the training with multiple GPUs. And we benchmarked our models with different strategies and GPU size types. And here in this graph, I'm essentially showing the cost of training each model in terms of training number of parameters. For example, for Inception V3, we had to train 24 million of parameters. And I'm comparing the cost of training a model like that over different T-flops available according to how we were distributing the training on the different GPUs. And we had our best performance by parallelizing over two V100s that are obviously the most powerful NVIDIA GPUs that I know at the moment, produced by NVIDIA. Okay, so finally, we proposed an explainable region proposer. So we focused our energies on developing interpretability tools alongside the development of the whole application. And next to the probability heat map of tumor regions that could be useful at testing time, we also tried to understand why a certain patch was predicted as positive rather than another patch, for example. So our first step was to apply previous research on the field by generating heat maps that could highlight the relevant input pixels in the image. We then expanded our research on the interpretability aspects, and this led us to quite interesting publications that I would like to present a bit more in detail. So one thing we realized is that interpretability outputs are not all the same, and some might be better than others in particular context. So we took into consideration the analysis of the field in terms of requirements. For essentially, we checked what clinicians want in terms of interpretability of deep learning models when applied to their pipelines. And this highlighted the need for understandable, clear, simple, and unambiguous explanation. So we kind of focused on the so-called human-centric explanations by putting the needs of the end users at the center of our development. And among the desirable properties, we found out an interpretability tools should be, as I said, understandable, clear, simple, and ambiguous, and that should be also actionable. So they should actually influence the decisions of the end users, for example, by asking for further checks, by rechecking eventually some areas in the image that they might have skipped. And so they actually they have a potential of translating into an action. And they should be generalizable to different models and architectures, and they should be consistent with the clinical values that clinicians follow. So concept attribution is one important interpretability framework that we developed within the project, where we explain the network output in terms of a concept. We essentially took inspiration from previous work on human-friendly interpretability. And the concept, this is essentially the concept activation vectors proposed by Kim, who proposed to interpret the network decisions in terms of binary concepts that can be either present or absent in the input image. So for example, here in this image of a lion, there are the concept brown and furry that I used to explain the classification. So brown and furry are concepts that are present, and the question is whether these concepts can explain the network classification, so whether these are relevant in the classification. And our idea, what we did essentially was extending this very powerful idea to measurable attributes that we can directly compute from the image. So we said we shouldn't have like binary labels. We should be able to actually directly segment a region where the lion is and compute measures representing the texture, the color, the shape of that region in the image where the lion is present. So going back to pathology, we took the grading system used to assess breast cancer tumor severity that I presented at the beginning of this talk. And we modeled one of the decision factors, namely the nuclei appearance in terms of the nuclei texture and size that we computed directly on the images. So we checked this nuclei pleomorphism that is within the grading guidelines, and we modeled it mathematically by evaluating the segmentation size of the nuclei and texture by computing texture descriptors that would tell us how does the nuclei appear within the image, if it has a coarse texture or if it has a smooth texture, for example. And we then use these concepts to produce explanations of the network decisions that showed somehow that the nuclei size and texture were actually relevant in intermediate network layers and that were used to make the decisions. So this was actually made to show that there is some clinical alignment between what the network is looking at and how the network is making decisions and what the clinicians would expect the network to be looking at. And just to give you a bit of a flavor of the details, I will go very quick in these slides, and you can ask me questions later on if you want to know more the details. But essentially, what we do is we first identify a concept of interest that describes the object that the network should classify, and we extract the descriptor for it. So for example, we could take object size, and we can compute the number of pixels within the segmentation of the objects as a measure of size. And we then pass this image to the CNN, and we extract internal activations after an aggregation operation, like special average, for example. And these activations are used they're computed for a set of images, and then they're used to learn a regression vector that essentially is a descriptor of the concept. And this regression is learned within the hidden space of a network, so we can do it for each layer. And we can use a last step that is called generalized saliency that would tell us essentially how much this vector representing the concept is relevant to determine the final output decision of the network. And this is done just by a directional derivative. So it's a simple computation of a derivative that then gives us a value that is the change in the network decision according to change of increasing values of the concept. So if we were increasing, for example, the size of the concept, what would we expect in the output of the network? Would we expect an increase of the probability or a decrease of the probability for that class, for example? In another work, we use then image scale as a concept to analyze its encoding in CNNs that are pre-trained in natural images. So one thing that is often done in medical imaging is to take state-of-the-art architectures that are pre-trained on natural images, and then to fine-tune, so to tune the parameters of these networks to be actually able to train and to classify medical images. And we find out that essentially scale information that is very relevant in medical imaging, because according to the size of the lesion, we take our decisions. And we found out that this information is present across almost all layers of networks pre-trained on natural images, and that invariance to scale is always only obtained at the last network layers. And we also saw that only 1% of the features within the network is actually enough to represent scale for multiple classes. So we exploited this information then in the application to histopathology, and we did this for essentially pruning out those network layers, pruning off those network layers that we don't need because they're learning the invariance to scale, and to then improve by keeping the scale covariant features, essentially to improve our results on the histopathology tasks. And I presented this work a week ago, essentially, at the International Workshop on Interpretability for Medical Imaging. So this is really fresh stuff. As for the future, one thing that we would like to try is to actively change the training of the network parameters by introducing the clinician's prior knowledge and feedback on the interpretability analysis. So the clinician, for example, could specify some important concepts to the CNN, asking the models to pay attention to the correct things and to discard what doesn't matter for the classification. So the clinician could ask the network to pay attention to the nuclei size, but not to learn the image domain. So to forget all of that source of heterogeneity in the data that causes problem when training deep learning models. So to forget the scanner type, the staining technique, and all these other details, while focusing on the important and relevant features that are used to make the classification also by humans. And this can be obtained technically by combining multitask and adversarial learning. So we can add this parallel branches, the learning of a list of control targets that can correspond to prior knowledge about our tasks, for example. And then we can combine this with adversarial learning to remove all of those representations that are far from the local minima of the adversarial task. So we could say you can use multitask learning to make those solutions that focus on the things you care about preferable, and use adversarial learning to make those solutions that you actually want to avoid, so to make them, to penalize them. And this is an idea that we have now as a preprint, and that we're working on it. So I'd like to give you some takeaways from the talk of today. Digital pathology took off in the last decade, generating massive data sets of gigapixel images. There are 1 billion pixels per image on average, and each image takes 2 to 3 gigabytes, and there could be up to 20 images per patient case, which makes up to massive data set. And deep learning can help the diagnostic procedure. It was shown to reduce disagreement, and it could also reduce the time for the analysis and the level of subjectivity in the analysis. However, changes in the acquisition procedure can generate heterogeneity in the data, and generalization should be improved by using several different data sets and reducing the domain shift among them. The algorithm should be brought to the data in the approach of evaluation as a service, So everything we developed should be actually in Docker containers and should be possible for the hospitals and private institutions to download them and train them. And development for AI in pathologists should be efficient, modular, portable, and interpretable. And it is with this final image of our application and the needs that we try to address that I would like now to officially open the floor for questions, if there are any.
Mara, many thanks for your lecture. It was really very interesting and with many, let's say, details important also for people for computer science. So many thanks.